Have you ever wondered how a single fertilized egg develops into an entire human body, or how children inherit traits from their parents? The answer lies in one extraordinary molecule, DNA. DNA is often called the blueprint of life because it stores all the genetic instructions needed to build, maintain, and reproduce every living organism. In today's video, we'll explore DNA from its basic building block to its famous double helix structure and understand why it's one of the most important molecules in biology. One fascinating fact about DNA is that every living organism have the same genetic language, whether it is bacteria or a human. All cells stores their genetic information in the form of DNA. Even more amazing thing is the genetic code is universally conserved, and this means the gene of one organism can be read by other organism. For example, scientists insert human insulin gene into bacterium and those bacterium produce human insulin, and vice versa. This simple principle forms the foundation of genetic engineering and biotechnology. Now the question comes, what are these DNA made up of? So what are the building blocks of DNA? So DNA is a long polymer of repeating units of nucleotides. Now what nucleotides compose of? Nucleotides are made up of three parts: the 5-carbon chain, deoxyribose sugar, and it is called deoxy because there is a missing OH group at the second carbon. The second component is phosphate group which connects neighboring nucleotides together, and the third component is nitrogenous base, and this is where the genetic information is stored. There are 4 different types of bases in this, which are adenine, guanine, cytosine, and thymine. These 4 are divided into 2 subtypes, which is purines and pyrimidines. Adenine and guanine are purines because they have double ring structure, and cytosine and thymine are called as pyrimidines because the structure have single ring structure. This distinction is important in DNA. A purine always pair with pyrimidine. This keeps the DNA at a constant width. So how nucleoside and nucleotides are formed? So what is nucleoside? Nitrogenous base is paired with sugar, deoxyribose sugar is called as nucleoside. When this nucleoside pair with phosphate group, it is known as nucleotide. And the bond in nucleoside is known as glycosidic bond, and the bond for the formation of nucleotide is known as phosphodiester bonds. And these bonds are formed by the removal of water. So now how is DNA chain is made? Now imagine connecting many nucleotides one after the other. So when the first nucleotide comes, it have a phosphate group from one nucleotide, forms a phosphodiester bond with the sugar of the next nucleotide. Specifically, the bond forms between the 3' hydroxyl group of one sugar and the 5' phosphate of the other sugar. This repeated linkage creates a sugar-phosphate backbone of DNA. Notice that the nitrogenous base projects outward while the backbone remains on the outside. This presence of phosphate and OH group gives a polarity to the DNA, which is 5' phosphate and 3' OH group, and by convention we write 5' to 3'. So now how is the double stranded DNA made? The double stranded is because of the 2 complementary strands which are anti-parallel. One strand runs from 5' to 3', other strand runs from 3' to 5', and the twist is right-handed helix. And while pairing, adenine on the first strand pair with thymine on the other strand. Similarly, thymine on the first strand pair with adenine on the other strand. In the same way, guanine on the first strand pair with cytosine on the other strand, and cytosine on the other strand pair with guanine on the other strand. This ensures that DNA remains stable and the distance between the strands remain constant. So now, how is this pairing happens during replication? Adenine, when paired with thymine, it forms with 2 hydrogen bonds, and when guanine pair with cytosine, it forms 3 hydrogen bond. And this is known as Watson-Crick base pairing rule. And how is the stability between the DNA strands are maintained? The bonds between the nucleotide bases are hydrogen bonds, which gives horizontal stability, and when it comes to vertical stability, it is due to base stacking interaction, which gives them vertical stability. These forces are driven by van der Waals forces. The flat bases stack tightly and stabilize the whole right-handed spiral, maintaining a constant width and stability to it. Now, how is DNA replicated? DNA replication is the process of copying DNA. So, before the cell divides, DNA helix unwinds and the 2 strands separate. Each original strand acts as a template, and an enzyme DNA polymerase reads the template to form the new strand. So, as described earlier, the template is being used and opposite strands are synthesized with the help of DNA polymerase, and with the base pairing rule of Watson and Crick, it matches the DNA of the template strand to add the new nucleotide. So the result out of it is high fidelity and preservation of genetic information. Now, what is the difference between eukaryotic and bacterial DNA? So, most eukaryotes, including humans, have linear DNA which are organized into chromosomes, while many bacteria have circular DNA which have no free ends, and it is highly stable and well suited for bacterial cells, which are called as plasmids. Plasmids, which can be used in biology to clone genes, produce recombinant proteins, and develop vaccines. Now, in a conclusion for DNA is a double helix structure made up of phosphate group, deoxyribose sugar, and nucleotides. The replication process is template directed to preserve the genetic information. It varies between eukaryote and prokaryote. Eukaryote have linear DNA, while prokaryote have circular DNA as plasmids, and plasmid can act as a powerful tool for gene transfer and biotechnology.